Hello everyone and welcome to today's webinar, Ultra-High-Content Imaging, a breath of fresh air for lung cancer research. I am Jennifer Woods of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit www.miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Telma Santos, Global Product Manager for Ultra-High-Content Imaging at Miltenyi Biotec. Dr. Santos, you may now begin your presentation.
Hello, and thank you for signing up for this webinar. In a previous webinar, my colleague Stefan Eulitz presented next-generation ultra-high-content analysis of the tumor immune microenvironment using mixed technology, and I would highly recommend you to see his presentation as well if you are interested in ultra-high-content imaging focused on the tumor microenvironment. In this presentation, I will focus on the potential of the mixed technology on lung cancer research, and I will give you some strategic approaches on the analysis of such data sets using our own QI tissue image analysis software. I hope you enjoy it. Let me start with lung cancer. Lung cancer is the most common type of cancer worldwide and can be divided in small cell lung cancer and non-small cell lung cancer. This one is the most common type of lung cancer and is the leading cause of cancer mortality worldwide. It can be divided in three subtypes: adenocarcinomas, squamous cell carcinomas, and large cell carcinomas. Lung adenocarcinomas and lung squamous cell carcinomas are the two major histological subtypes, accounting for almost 70% of the total cases of lung cancer. What differentiates them mainly is where they begin. The adenocarcinoma begins in the cells in the glands located in the outer part of the lung, whereas the majority of cases of squamous cell carcinoma starts in the center of the lung. The current treatments for lung cancer include surgery, chemotherapy, radiation, targeted therapies, and immunotherapy, and have improved a lot in the last few years. Immunotherapy is a quite exciting new kind of therapy because it uses your immune cells to fight the tumor cells. It is quite promising and often shows long-term clinical benefits and even in some cases a complete recovery. But the major limitations include a very high variability in response to treatment and a difficulty in proper qualification for therapy. So we need to have deeper insights in the organization and content of the tumors. And this is not only to develop new kinds of immunotherapy, but also to be able to really identify which kind of patients would be better suited for which kind of therapy. And why do we need such deeper insights? So here you have a depiction of a heterogeneous tumor microenvironment. A lot of tumor microenvironments are like this, very heterogeneous. You don't really know what's in there. And the thing is that if you look at the regular imaging, a regular microscopy image, you normally would not see so much because you are limited in the amount of markers that help you recognize the different kind of cells. And here I have some examples. If you go for tumor or for the immune cells or for some other cell types that are often found in these tumor microenvironments, you need a lot of markers just to characterize them. And this is known. This is not something new. So imagine if you need to know more or to even try to figure out more to define new markers, you would need more than 50 easily. And the Maxima imaging system is actually the only a system that can do so many markers, hundreds of markers in one sample. MACSima imaging system was developed by Miltenyi Biotec and uses the mixed technology to achieve such high numbers of markers in a sample. The mixed technology stands for Maxima Imaging Cyclic Staining and is quite simple to understand actually. The Maxima is an automated system, meaning that you just need to put in your sample and your antibodies inside, and the machine will do the rest. It will basically take the sample and stain it with an antibody, image it, and erase the signal, and therefore can stain it with other antibodies, image it, and erase it, and repeat this cycle as often as you need to. And this way we can have hundreds of markers in a sample. What we did was not to just create a system, we created an entire platform. So with the Maxima Imaging Platform, you do not just have a fully automated instrument that can perform such amazing kind of experiments, but you have also everything you need for the full experimental plan. So you have sample carriers that can adapt for different kind of samples. You have a huge range of validated antibodies, and we have an amazing image analysis software, which I will present a bit later on because this is how I did all my analysis. This particular experiment was done by imaging more than 150 markers in a sample of lung adenocarcinoma and a sample of lung squamous cell carcinoma. And here you have a depiction a bit of some of the markers that were used, and you can see how great it is to see so many different markers of different cells in one single sample. I will start this part of the presentation by not going into real numerical or graphical analysis, but really to just show you the markers, the more common markers that are used for the identification of cell population, and see how they are organized between the lung adenocarcinoma and the squamous cell carcinoma to see really the difference between these two kind of samples. I will start to introduce you just an overview of how we start the experiment. So basically, in this case, these were sections were on a microscopic slide. For that, we would use then these sample carrier, the Maxwell 4 imaging frame. So we have different sizes, but this one has 4 cavities basically. And you mount your microscopic slide here on our imaging frame, and then it will be able, the Maxima will be able to stain then because it has the cavity necessary for it. And we had a sample of adenocarcinoma in this well and a sample of squamous cell carcinoma in this well, basically. And the antibodies was the same mix. It was the same use for both. They were all processed at the same time. When you start the Maxima, what it does is that it does an overview image of your well, so you can see your sample, and then you can define the area that you want to image. And this is the reason why you'll see that the two samples have a different format, because this is the format that was selected when the experiment was done. So I will start with the general overview of the sample. So basically what I made here is a composite with 7 markers to identify 6 most commonly described cell population in this kind of tumor samples. So basically on the left, you'll always see the adenocarcinoma, and on the right, the squamous cell carcinoma. In green, you can see the tumor cells who are stained here by cytokeratin. You can see the T cells stained by CD3, the B cells by CD20, the myeloid cells stained by CD163 and CD15. You see some endothelial cells, so for the blood vessels with CD202B, and you can see some fibroblasts with the vimentin in gray. And what you can see here immediately is that it's difficult to make sense of it immediately, but first you can see directly that they have a complete different kind of organization. In here, most of the cells seem to be cancer cells, and you have some stroma area here, and you see some infiltration from T cells, and you see that the rest is basically myeloid cells, fibroblasts, and some blood vessels. When you look at the squamous cell carcinoma, you see that the proportion of tumor cells or tumor positive cells are not so high. It would be more on a 50-50 level. You barely see any infiltration of T cells there. You can see that we have these very weird regions that seem to be necrotic areas because the shape and the intensity of the signal here is very different from the rest. You see that in the stroma area, you have mainly fibroblast and myeloid cells and almost no blood vessel marker here. So I would say that here we do not have infiltration apparently. But now focusing only on the T cells, so this helps visualize it better. So now we can clearly see that there are way more T cells in the adenocarcinoma, and it doesn't seem to have much in the squamous cell carcinoma. But let's have a closer look. Yeah, so here looking at the markers, we can clearly see in this region where we have more T cells that, so first CD3 is more common T cell marker, so it will be in all T cells. The CD4 is just on T helper cells. And the CD8 is more in cytotoxic T cells. CD8 is normally associated with a better prognostic. Here we can see that the majority in the adenocarcinoma of the T cells are CD4, but we can see some CD8 positive cells. In the squamous cell carcinoma, though, we can see that there is barely any signal. We see some speckles, but I would not take this as positive cells. So I would say that this particular sample has no infiltration. Now let's have a look at the B cell marker. So CD20 is a common B cell marker. So first on the adenocarcinoma here, the signal seems a bit weird, not a lot. It seems almost that it's speckled, so I'm not sure how it is. When you look at the B cells, it has a very weird pattern. It seems that we have signal in the nuclei of tumor cells. So this is very weird, first because it is not replicated in the other sample and it is the same antibody from the same mixture and the samples have been handled the same way. So this is very curious. Of course, you cannot take any real conclusion out of it because further investigation would be needed for it, but I still wanted to show it because I found this quite interesting, and also for the fact that we have here direct comparison of the markers. But let's take a closer look. So here, when we look at it more in depth, we can see that actually here we have real signal. It's just that there's not many B cells in this sample. And here again, we can see this very interesting pattern. And again, this is very curious because it's only in the tumor cells. It's not on the other cells. And here we actually can see a real, well, an expected pattern of this antibody because it stains where it's supposed to stain. So I do not think that there's a real problem with antibody, but we should still, this would need to be figured out in other samples to see if this happens again or if it's just a one time or, but it's still very interesting as an observation. Now I'm here showing you some markers that are used for myeloid cells. So here there are a couple, so it's can be a bit overwhelming, but all in all, what you can see is that in the adenocarcinoma, it seems that they are a bit distributed. You can see them evenly distributed a bit everywhere, not really clear pattern. We see that the HLA is more visible here in some places here, but the rest seems to be a bit well distributed. When you look at the squamous cell carcinoma, you see that we see a very big amount of signal of CD15 in these necrotic areas, which could make sense. And actually, you see that in the stroma area, you have the rest of the markers mainly there. So again, based on the other observations before, we could see that the stroma area of the squamous cell carcinoma seem to be constituted mainly by fibroblasts and myeloid cells. So it makes kind of sense, but we can see some CD14 areas here around the tumor cells. So this can be very interesting as well. But all in all, we see how different they are organized. So here I'm looking mainly at the fibroblast. So I used vimentin as a marker generally for general fibroblasts, and the other ones as activating markers. So you can see that first of all they overlap the activating markers. So actin is known to be an activating marker. Caldesmon and myosin can be associated with it because it kind of is into the transition from the state of activation. And so here what you can see is that we can clearly see some specific areas where there seem to be some fibroblast activation. When I say activation, it is normally in support, in favor of the tumor against infiltration of immune cells. So that's not very good for prognosis. We can see here that in this area, which coincidentally we know have some infiltration from the other images, so it can be kind of a reaction of tumor cells to prevent infiltration to go further on. And here you can also see, interestingly, that these activation areas are very at the border between the tumor cell and the stroma area. So it could be supporting the growth of the tumor cells. Assessing the tumor proliferation state is also very important when looking at tumor samples. So here I used Ki67 and PCNA to look at the proliferative state of these tumor cells. So we can see that all in all, both samples seem to be quite active. We can see that the adenocarcinoma, there are some areas that seems way more active than others. And we saw before that there was some T-cell infiltration as well. Here it might be that it means that they're fighting actively the tumor, and that is good. In the squamous cell carcinoma, basically, if we forget the necrotic areas, we kind of see that in all the tumor cells here, so the non-necrotic tumor cells, you can see basically they're all in a highly proliferation state. So this tumor doesn't look good. So here I wanted to check the environment of the tumor sample. So basically, PD-1 blockade is one kind of therapy that is commonly used in tumor therapy, and what it means is that basically when we have PD-1 and PD-L1 and PD-L2, which are the ligands, present in a sample, this would suggest that the sample has an immunosuppressive environment, meaning that it's not good for cells, for immune cells to infiltrate to fight the cancer. And here we can see clearly that there is the presence of these markers in the squamous cell carcinoma, but not really in the adenocarcinoma, which could be why there is no really T-cell infiltration, for example, in this sample. So now I would like to focus mainly on some strategies for the analysis of such samples. So as you might understand or might imagine, this kind of data can be very overwhelming. It is a lot of markers. It's a high level of content within one sample only, and it can be very overwhelming to figure out what we really want to extract from it. So you can start your experiment with one question in mind, but you have such a high amount of data inside that it's not uncommon that you would go back to your data set to find out other conclusions out of it, because it is so rich that it is not meant just to answer one quick question. So in this first part, I will focus on the lung adenocarcinoma, and with some ideas for you how you could handle this kind of analysis and how you would display such kind of results. I will start though by introducing our software here, because when acquiring a Maxima, you will get the QI tissue image analysis software, and it's a very complete and very nice tool to use for ultra-high content image analysis. It has been developed for this kind of data set especially, and it offers a variety of features. And here I just want to show you a couple of them for you to have an idea where the software looks like. So basically, you can easily see all your markers from your run. So this is a very nice way to visualize all your data. There is also segmentation integrated. So you can, with a click in a couple of parameters, just start and have directly the segmentation. And segmentation is crucial to define the cell nucleus and the cell boundaries to give an identity to a certain area, to a cell identity, so then you can correlate cells with markers, expression and location, etc. With this software, it is highly interactive. So when you do, for example, gating strategies, which our flow cytometrists are very familiar with, you can do it like you are used to, but the plus here is that you actually see which cells you are selecting. You see them lighting up on the tissue, and you can also do gating within the tissue sample. So you can select the cells and you can see them light up in your graph as well. And of course, there's integrated a variety of visualization modes for your data to better explain the data that you're extracting. So let me start with the first strategy. Based on the observation that was made by looking through the most common cell markers as we did before, I concluded that the adenocarcinoma sample was mainly constituted by tumor cells, T cells, fibroblasts, and macrophages. So in this strategy, I decided what kind of cells I wanted to look into and how I would group them. So first, I started by doing a very simplistic gating strategy. So this can be, of course, more complex depending on where you come from and what your objectives of these samples are. But this, I wanted to provide an easy to grasp gating method. And for that, I started by selecting the tumor cells versus the T cells, and then I said that the rest of the cells would then be fibroblasts and macrophages. Then I gated on a histogram the fibroblasts based on the vimentin, and then used the rest of the cells as macrophages. So as I said, it's a very simplistic. There's certain ways to do it better, of course, but this is a very easy way to explain it. After doing this kind of gating strategy, you could actually see the cells you are selecting. So I could directly see the cells I selected and assign them a color and the name, of course, and see them on the image. You can also get the information about the number and the percentage of cells as well. Gating strategy, what I wanted to do with it was to define how these different populations were organized in relation to each other. So what I did was I created distance maps. So with these distance maps, you get basically an image where the higher the gray value, the higher the distance from the cells of interest you are. So I created 4 of these maps and then plotted them against the cells as well, to define how they were organized, which ones were closer together and which ones were furthest away together. And based on this, I could see that the macrophages seemed to be closer to the tumor area, whereas the T cells furthest away, and the fibroblasts seemed to be intermediate. So this could suggest that actually in this adenocarcinoma, the cells are not managing, the T cells are not managing to infiltrate as much as we could, as much as we wanted to. And that actually maybe the fibroblasts are there to preventing them to go, since they don't manage to pass them. As a second strategy here, I wanted to include a quite different feature that you wouldn't have by doing flow cytometry, which is really to look at the shape of the cells you're looking at. So when you look at the, so here you have the DAPI in gray with the green being the cytokeratin, so the tumor cells. But you can also see just the DAPI image here. And you can see clearly that you can identify the non-tumor cells just by looking at the nucleus, at least some areas. The nuclei of the tumor cell is very irregular and is even has lower intensity. These ones seem to have holes. They seem to be hollow. So this is a feature that is actually extracted from our data, so that can be plotted against each other. So I decided here to plot the nucleus roundness versus the cytokeratin to really create 4 kind of groups. So the round and cytokeratin negative cells, the round and cytokeratin positive cells, the irregular versus the irregular and cytokeratin negative, and the irregular and the cytokeratin positive cells, and see if I could see the differences between these four. So I decided to plot all the markers I had in this run in a heat map to just have a better overlook and then have the possibility to go further into it. But here I will just stay here. This is a strategy for you to imagine how to handle this kind of data. So first of all, I could see that the round and healthy, so the round and cytokeratin negative cells seem to have most of the immune cell markers, such as the CD2, CD5, CD4, CD3, and CD8 also. So this could be seen here actually quite nicely. We could see that the cytokeratin negative cell had levels of CD66E and CD162, which are related to a kind of inflammation state of the immune cell. So that's also very interesting. We could see that CD147 was more present in the cytokeratin positive cells, but it's slightly bit more in the brown one, so it might be that they're not directly already final tumor cells, but on the verge or contacting with them. And CD147 is actually a rising star in the target research because it's thought to be very good. That could be actually a potential nice drug target. It is involved in the extracellular proteinase inducement. So it induces extracellular proteinases, which helps the tumor grow in theory. You can see also here the cytokeratins nicely differentially expressed, and you could see that the cytokeratin positive cells seem to have more HSP70 and PI3K, which is related with a higher activity, which is common in proliferating cells. So these were my two strategies for the lung adenocarcinoma sample, and here I would like to focus more on the squamous cell carcinoma. Since they had such different kind of organization, I thought they were perfect examples to have different strategies associated with them. So for the squamous cell carcinoma, first I wanted to show again the different organization of this tumor. Basically, here you can see that I have 4 markers plus the DAPI showing the tumor cells in green with the cytokeratin, the vimentin for the fibroblast, and the CD163 and the CD68 for the myeloid cells, and you can clearly see that you can distinguish 3 different areas. So we have the stromal area, which is with non-tumor cells, we have the tumor cells, and then we have these necrotic areas who have some tumor cell markers, who have some myeloid cell markers, but seem to be really not that healthy as well, and do not have proliferation markers, for example, and have these very weird speckles. What I decided to do here, was to first do an image gating strategy. So I really like just to draw these regions and selected them as the necrotic areas. And then I quickly just gated the tumor cell positives with cytokeratin versus the rest, and to separate them in these 3 major categories. And then I decided to go for a overview look, a screening. So I call it screening method every time that I am doing a heat map, because I found the heat map the best way to have a overlook of what's going on, especially when you're comparing different cells. So for example, in this case, you could see that the necrotic areas seem to have a lower amount of cytoskeletal proteins compared to the tumor, which would make sense. If they're dying, they're not reproducing, they're not making new building blocks for the new cells. We have also galectins who are lower, and they are associated with proliferative cell as well. So it makes sense that they're more they are higher in the tumor, and we know that this tumor selection, because we excluded the necrotic areas, right, that are more proliferative, and you can actually see this clearly in the Ki67 and PCNA area. We could see also that high levels of 66 variants were present in a necrotic area, which is often associated with inflammation. So it would make sense that the necrotic area would be highly inflamed. And we can also see that there are high levels of CD15 and amyloid A. They're suggesting that these necrotic areas have high inflammation going on and some recycling of the membranes, and there might be some digesting of the necrotic cells there. Based on the same guiding strategy of before, so by the image gate of the necrotic areas and the tumor versus non-tumor areas gating here, based on that particular gating strategy, I decided here for the second strategy to just do a distance map of the tumor cells. So basically what I wanted to show here is to define which are the cells who are more in contact with the tumor versus the cells who are further away. And once you create this kind of map, so what you can do is that you can plot it. So for example, here I have the intensity of the pixels, and I can plot the ones who are lower intensity means that they are closer to my tumor cells, and the ones who are higher intensity, that will be here, for example, will be furthest away. And when you plot them, here I use the same coloring, and here you can see them plotted, so you can see them selected here in the sample. You can see that the pink ones are the ones really surrounding the tumor areas, and then the green ones are really in these areas where they're furthest away from all kinds of cancer cells. And here you could see then that they showed to have different markers expressed. Actually, you could see that, for example, here we have some CD38 and CD147 very active. CD38 interacts with extracellular proteins, and as I said before, CD147 is an extracellular proteinase inducer, so it can explain that helps. These are the cells that helps the tumor cell expand. We see some CD66 here, so correlated with inflammation from the cells who are not tumor cells, so they can be in an inflammatory state. We also have JAK1 here and SIK here, also related to inflammation. And here we actually can see a nice gradient where we see that there is more tubulin, so more alpha tubulin close to the tumor region, and less as you go further away. This can be a relation to the activated state, so the proliferating state that we can see, that closer to the tumor there is more active cells who are either moving a lot or dividing a lot, and therefore they would need more tubulin. I would like to now present a very quick last strategy. So of course you can do all these manual gatings as well, just to choose the cells that you already know are there, that they exist, and observe how they organize between each other and what kind of content they have in it. But when you do not know and you want to figure out if there are some different groups of subtypes of a cell, for example, in this case here, subtypes of tumor cells, what you can go for is a more unbiased strategy where you would look at the different clusters of the tumor cells based on the markers that you wished for. So here, what I did was that I did a k-means clustering of the tumor cells only, in the hope to get some interesting clusters to identify and maybe define new subtypes of the cancer, of the tumor cells. And basically after plotting it, I also did a DZNE plot. These kind of plots helps you to have an overview of how the cells are organized in between each other. And here you can see clearly where these cluster falls in. So cluster 1 here, cluster 2 here, cluster 3 here, and so on and so forth. And then what I did is, like, I plotted these clusters in a heat map also again to have an overview. One example here is that you see that cluster 1, 4, and 5, which are the biggest ones, do not seem to have stand-up standouts, but it might be to the fact that these two clusters, because they're very small, stand out better, and then kind of make the other differences not as visible. So in a case here, we could do is then just plot these three clusters between each other, ignoring these two, to see the big difference between them. Of course, when you have smaller clusters, chances are that you're going to see bigger differences. And here, as you can see some of the cells, the blue cells here, and the green ones are very little amount. You can barely see them. So this might be just a very, very specific kind of cells. And in this case here, what I could see is that cytokeratin 14 was high only in this kind of cluster, as well as podoplanin. So this is quite interesting. So I do not know. So cytokeratin 14 would be more in epithelial cell. Podoplanin could be also. So this is interesting. Now we have in the cluster 2 amyloid A, who seems to get higher, and GFAP, for example, which is quite interesting because these are known normally to be neuronal markers. In this case, they are not. So because they're tumor cells in the lung. So this is something that could be looked at into more deeply. And here you can see that these clusters seem to have the highest amount of all the cytokeratins compared to the three clusters, and these are only tumor cells. So this is quite example. We could have a further look into these clusters and dig into a bit more. But these are general strategies that I wanted to present to you to get an overview of what you can do when you have this data, at least the first steps to start understanding what your data has. So basically, I hope that you enjoyed this presentation. I wanted to show again the potential when it comes to ultra-high content data that can only be generated with the Maxima at this level with hundreds of markers. You can make such nice presentations and content using our QI tissue software for the analysis of these data sets. That has already very many features that are needed for this kind of analysis. So just as a summary, I would like to say again that the MACSima Imaging Platform really allows for an unprecedented number of markers in a single sample, and it's really breathtaking when you look at all the markers that you can see in one sample only. The ultra-high content imaging opens the door to a variety of analysis approaches. So I gave a couple of examples here, but really the sky is your limit. It depends really what you want to find out, or sometimes you don't even know what you want to find out, and you can find out great things just with these rich data sets. As I said, it won't be uncommon to go back to your experiment and have other outputs out of it once you are done with the research question that you had before when you did your experiment. You can always focus initially on the T cells and then go on the tumor cells and so on. The way you look at your data can change, and it can really give you different outputs. This software allows for multiple analysis strategies, very complete and very convenient to use and easy to use and elegant. And as you can see, you can get quite a lot out of it. So just wanted to finish by showing again this nice animation where you can see all the markers. I would like to take this moment to thank everyone that is involved in this, such as our researcher, our application development team, and also our developers for QI tissue. This is really, this has been great, and we are making great products here to really enable research to another level. I would like to now also thank you for your attention, and I'll be happy to take some questions. Thank you.
Thank you, Dr. Santos, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is there a way to check histology in bright field?
Yes, so currently the Maxima does not have a bright field image generated. What we normally use is the DAPI signal to check the way the tissue is. So currently we cannot do bright field.
Okay, thank you, Dr. Santos. Our next question is it possible to have an idea of the computation time for calculating distances, defining areas, etc., plus experimental time?
That is a great question, but can be also a bit complicated to answer. So basically, I believe that what takes most time is looking at your data set, especially when you have such huge data sets. It takes some time to look at it, to really see what worked, what didn't work, what is the markers that already have my interest and everything, just to go through it and have a strategy in your mind already. So what is my aim? What do I need to analyze what is my current interest? Because as I said, you can have really different outputs depending on how you look at your data set. The part that I showed here, once I decided which strategy I wanted to go, was very, very fast. The generation of maps is very fast in QI tissue, for example. So it can take time, but it's not decisive. Is not from the software. It is not from the Maxima itself. The time that will take, what will take more time to do is to define what you want. Once it's quite fast. The software is fast, and also depending on how big is your data set, your computer can be a bit faster or not. But in general, for sure, it's really fast. So so the image there of the distances was generated in 3 seconds.
Thank you, Dr. Santos. Next question. Do you use markers or nuclei shape to define necrotic areas?
So in this one, particularly, what I did was just checking at the nuclei because it was quite strong. So all these very pointy and very strong signal. I didn't check specifically for markers.
Okay. Let's see. Is it only possible to look at ROIs rather than full tissue sections?
So you define your ROI. It will depend on your choice. So if you want to select the full section, you can, as long as it fits into our disposable. So our biggest disposable is the Maxwell one, and you can have most of the portion of your microscope slide there. But of course, you will have to consider that this will take more time because of all the imaging processing and also the experiment to be done. But it's possible, yes.
Okay, thank you, Dr. Santos. Next question. What is the max size of a scanning area, and what is the speed of scanning and resolution of the images?
So as I just said, if you use our Maxwell 1, which is the one with the bigger area, you could, in theory, do a bit more than 900 square millimeters. But of course, you have to consider the parts that it will take more time because you will have to acquire multiple field of views. Our best resolution currently is a bit more than 400 nanometers with one of our objectives. And the time will really depend on a lot your configuration of your experiment. So the shortest possible cycle could be around 30 minutes. but it will depend on how many antibodies you have in one cycle, how many field of use you have. So it depends on a variety of parameters.
Okay, great. Thank you so much for that answer. Next question here. How many antibodies are used per cycle, and what chemistry is used for signal removal?
So currently we can use up to 3 antibodies in one cycle, plus the DAPI signal. And in terms of signal erasure, we have, so on one hand, we use photobleaching, and that is why we have the fluorophores that we have. And we can also do another mechanisms based on our Realis technology that we developed at Miltenyi, and this is a controlled fluorochrome release of the antibody. Of the fluorophore, sorry.
Okay, thank you. And next question. Let's see. Are the antibodies used in a stain directly conjugated? Is amplification used? And are those antibodies proprietary or common clones?
So the antibodies we used are primarily conjugated antibodies. So there's no signal amplification, and we so when in our R&D we mostly use Miltenyi antibodies, of course, who are mix validated. But you can use any antibody as long as it fulfills some requirements, which is the fact that it can be stained in 10 minutes, because that's the process in the Maxima, and it can be and it is conjugated to APC, FITC, or PE, because these are the fluorophores for which our photobleaching is currently set up for.
Okay, thank you. Next question. Are these 2D whole slide images, or do you have to select an ROI before image acquisition?
So when you start the software, the Maxima will do an overview scan of the whole well, and then there you can select one ROI or multiple ROIs or a very big region of interest. This is totally your choice. In this particular case, also I showed in one of the slides, it was just one region. So in the Adenocarcinoma, we had about 18 field of views, and in the Squamous cell carcinoma, it was about 16 field of views.
We've got time for a few more questions here. Our next question, what is the resolution that can be achieved with this technology?
So we have two objectives. One that can image through the microscope slides, which can achieve around 600 nanometers, and one for very thin glass who can achieve around 400 nanometers.
Okay, thank you, Dr. Santos. Our next question. I would like to know if only antibodies validated by Miltenyi can be used with the machine, and if this is the case, is there a plan to expand to non-optimized antibodies?
So of course we will always say that validated antibodies are a bit better, and I'm sure everyone understands the struggle of validating a certain antibody. But the Maxima is an open system, so as I said, you just have to fulfill a few requirements. You would just have to get the antibody that you want conjugated to one of the fluorophores, and make sure on your hand that you have tested it for stain duration of 10 minutes and define the titer yourself. But after that, you could just have it on your antibody plate, for example, and the Maxima will make sure it's used in the cycle. So it's not close to only Miltenyi validated antibodies.
Okay, thank you, Dr. Santos. Looks like we have one more question here. Can you quantify the signal intensity as well?
So yes, of course, the images are all acquired and processed, and then once you have an imaging software program that can open TIFF files, which is virtually all image programs, you will have there the signal quantification with your gray values.
All right. Well, thank you, Dr. Santos. Do you have any final comments for our audience before we go?
Yes, so I would like to say that I hope first that everyone enjoyed the presentation and I could convey a bit more information about what our platform can do, not only in terms of the technology itself, but also show you some examples of how you can handle such kind of data and how you can think about the data that you want to extract from your experiments. And I'll hope that you could see also that with QI tissue, you could achieve a lot of interesting outputs. Of course, there's a lot more, and again, it always depends on what kind of questions you have and what you want to answer. It might be then that you can get new questions just by looking at your data. I just also wanted to say that there's always more to come, that you can always follow us on our social media and check our websites for the latest news. And of course, if you have more interest, do not hesitate in contacting us for more questions regarding the Maxima platform. So everything from the instrument, from everything we offer. Thank you again for participating in the webinar, and I hope to see you in the next one.
Well, thank you again, Dr. Santos, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.